Hello, my name is Courtney and I'm a student in the Marine Genomics Lab at Stellenbosch University. My research focuses on metabarcoding of estuaries as a tool for biodiversity monitoring. Biodiversity monitoring or biomonitoring is essential to protect, preserve and restore ecosystems. Traditionally, biomonitoring has relied on morphology-based identification, but this proves time-consuming and often requires taxonomic expertise. In addition, it usually focuses on larger macroinvertebrates as they're easier to catch and identify. An alternative approach for biomonitoring is metabarcoding of environmental DNA. It has the advantage of efficient sampling, does not require taxonomic expertise, and is non-invasive. Briefly, the metabarcoding pipeline works on that organisms shed DNA into their environments, which we term eDNA. We can sample this eDNA by taking a sample of the substrate, such as water or sediment. We can then perform DNA extraction on the substrate and send the DNA for sequencing. Once sequenced, we can resolve the sequences into OTUs, which are operational taxonomic units. They're basically groups of very similar sequences. The OTUs are then queried against databases to give the closest identity match to a particular group. This provides a snapshot of the community in time and space. So my research focuses on sampling 6 Zostera capensis seagrass estuaries along the coastline of South Africa, with 2 estuaries per biogeographic region, namely subtropical, warm temperate and cool temperate. From each estuary, water eDNA and sediment eDNA were collected, with sediment being collected from intertidal unvegetated, intertidal vegetated and subtidal vegetated seagrass beds. In addition, I collected macroinvertebrates from the seagrass beds for morphology-based identification and DNA barcoding. So the aims of my research were as follows. Firstly, to compare the OTUs returned by metabarcoding mediums. Secondly, to explore the communities captured by metabarcoding mediums. To determine whether metabarcoding reveals biogeographical patterns. And to compare the results of morphology-based identification and metabarcoding. So firstly, we can compare the OTUs returned by mediums with pie charts of the number of OTUs with lowest taxonomic identification at kingdom through to species for the 4 metabarcoding mediums. So we can see that most OTUs were only identified at the kingdom level, with very few identified to genus and species. And this may be due to the fact that barcode databases, particularly in the marine field, are completely insufficient. And in order to have a match, you have to have a very high similarity. What we can see, though, is that for the 4 mediums, this trend is the same. Here we have Venn diagrams of sampling mediums OTUs for sites. So here is the sediment samples and then the blue is the water samples. So we can see that there is an overlap, particularly between the sediment samples, and that for all the sites, water tends to have a large proportion that is unique. To explore that further, here's a box plot of the number of OTUs for the 4 mediums at 6 sites. And to determine whether the mediums returned equal numbers of OTUs, I performed a Kruskal-Wallis chi-squared and found no significant difference. Therefore, the null hypothesis is not rejected. Next, we have a box plot of the number of unique OTUs for sampling mediums. To test the hypothesis that mediums returned equal numbers of unique OTUs, I performed an ANOVA and found a significant difference between the number of unique OTUs, in particular between water and intertidal vegetated. As such, the alternative hypothesis is that water returned more unique OTUs than sediment mediums, which we saw in the Venn diagrams. Next, we can explore the communities captured by mediums. So here we have a stacked bar plot of the number of reads, which is the number of sequences, for the phylums collected by metabarcoding, for sampling sites, and their mediums. So what I'd like to iterate here is that number of reads does not necessarily equal abundance, because you could have a bigger organism which just sheds more DNA than a smaller organism. But what we can see is that there tends to be a trend that sediment samples have more reads than water samples, although not at breeder. But this isn't very informative. This is more informative. So this is proportional. Here we have the percentage of total reads. Off the bat, we can see that there is some strong community structuring between mediums, especially at breeder. But generally, the sediment samples tend to have a more similar community structure than their water counterparts. Now there's lots we can read off this graph, but just two things I want to point out. Firstly, the water samples have a high proportion of ochrophyta, which is the brown algae. And brown algae is water dwelling, which could explain why it's so prominent in water. In addition, the sediment samples have a high proportion of Annelida, which burrows in the sediment, which could explain why it's so prevalent in the sediment samples. Now, something that we can test is whether the communities differ significantly between the mediums, so between water and sediment mediums. In addition, we can determine whether communities differ between biogeographic regions. So here we have an NMDS plot of Czachur distances for sites OTUs grouped by biogeographic region and sampling medium. So in orange is subtropical, green is warm temperate, and purple is cool temperate, and we can immediately see strong clustering there. In addition, the sediment samples are all grouped together, quite distinct from the water sample. But now we want to test, did the mediums capture similar communities? And a two-way Permanova found no significant difference, but this could be compounded by the sediment samples. So pairwise comparisons offer a more in-depth analysis. Water compared to sediment are in blue, and sediment compared to sediment are in red. And we see that sediment had much higher p-values and are far more similar compared to water. As such, water captured a distinct community to sediment mediums. But now we ask the question, what are the OTUs or the taxa responsible for the dissimilarity between the mediums? To answer that, here's a correspondence analysis scatter plot of the OTUs at phylum level that contributed to a quarter of the dissimilarity between mediums. So here are the sediment samples and here are the water samples. And we can note two things. Earlier we pointed out that ochrophyta were very prevalent in water, and we can see how they tend to cluster around water. In addition, Annelida were very prevalent in sediment, and it also tends to cluster around sediment. So what we can draw from this are the OTUs, or the phylum, that tend to cluster in water or sediment samples and will therefore be picked up more easily in those mediums. So next we can investigate whether the clustering is significant for biogeographic regions. So the two-way Permanova found that there was a significant difference, very significant. In particular, cool temperate was more similar to warm temperate than subtropical, which we'd expect given that cool temperate and subtropical are on opposite sides of the coastline. As such, sites within biogeographic regions were more similar than sites between regions, and there was biogeographical clustering. In addition, the two-way Permanova found that there was no significant interaction between medium and region. Now once again we can ask, what are the OTUs or the taxa that are responsible for this biogeographic clustering? So here's another correspondence analysis scatter plot of the OTUs at phylum level, contributing to a quarter of the dissimilarity between the regions. Now this kind of plot is useful because we can draw from it the OTUs or taxa that are possibly sensitive to the environmental conditions if they are creating such dissimilarity, and they could therefore be used as possible bioindicator species. Now here we only have it at phylum level, but some of these did go down to genus and species level, so they would be interesting to watch and monitor. Next, I wanted to compare the results of morphology-based identification and meta-barcoding. So here we have Venn diagrams of the taxa taxa at genus level that were identified through morphology and metabarcoding. So we can see there's very little overlap between the taxa that were identified. In addition, morphology identified far more taxa at genus level than metabarcoding. However, on the right, we have the phyla that were identified through morphology and metabarcoding. So, metabarcoding identified far more phyla, but had a far lower taxonomic resolution, whereas morphology identified only a few phyla that were larger macroinvertebrates, but had finer taxonomic resolution. They therefore identified different taxa. So the conclusions and takeaways. Firstly, water and sediment mediums returned unique communities, so sample multiple mediums for a full community assessment. Secondly, metabarcoding revealed biogeographic patterns, and from this we can determine taxa that contribute to biogeographical structuring, and these could be possible bioindicator species. Third, metabarcoding identified a broad range of organisms as well as unique phyla, while morphology-based identification resolved more taxa to genus level. As such, supplement morphology-based identification with metabarcoding. Finally, a more robust database will resolve OTUs to finer taxonomic resolution. So barcode, barcode, barcode. If we can strengthen the database, then we can have a higher percentage hits, which results in a finer taxonomic resolution. Thank you.